And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Example of speciation over the past 12,000 years.
Yeah, Jackson, that's really going to excite 100% of guests. That
and then they run. They always run.
That's what I was going to say. Yeah.
No, it's a fascinating subject. Why are you running?
I had a, why are you running? Why are you running? I had an uncle that asked me about some of the...
You guys live.
Okay, awesome. Looks like we are live, and we are live tonight with a debate to remember, I'm sure. Both Dr. Kevin Anderson and Jackson Weed have been generous enough to give us their time for this much anticipated and much needed debate. The topic of tonight's debate is whether or not universal common descent is evidence-based. One quick announcement before I introduce everybody tonight. Don't forget to be here this Thursday night, same time, 8:30 EST, for another highly anticipated debate between Dr. Dino Ken Hoven and Dr. Dan Cardinale on macroevolution versus microevolution. So lots to look forward to this week. I also want to thank the moderators for the night, Guzman and Erica, for joining us and helping us out. So any words from the moderators? Thanks again for helping out here.
Oh, we're just glad to be here. I know Guzman's excited. I'm excited. I know both of our debaters today through their work or directly. I will shout out for Logical, Plausible, Probable, who is hosting an after show on his channel, and then I am also hosting an after show on my channel. So depending, they both offer different perspectives on what's going to be on the conversation. The dialogue is going to be tonight. So I would like to invite everyone to attend one or both or whatever you would like to do.
Awesome. That's it. The after shows are always a lot of fun. So be sure to check out Guts at Gibbons and Logical, Plausible, Probable. So, and Brother Guzman, I want to give you a shout out as well for helping to arrange this debate and making it happen, man. Any words from you?
Well, no, not really. Just thanks, Standing, for giving us the opportunity to get Kevin Anderson and Jackson Weed together. This is going to be a really good debate. I'm going to have fun tonight.
Awesome. Well, I've got to say, we've got a large audience tonight in the live chat, so that means there's going to be a lot of great question, and so the help is greatly appreciated. Now, as always, what we'll do before we go over the structure and kind of get the debate going itself, I want to give both Jackson Wheat and Kevin Anderson just a few moments to give an introduction as to who they are, maybe some background, what they've been up to, really just anything they want to say in order to break the ice. We can start with you, Jackson. The floor is yours.
I'm nobody. Don't pay any attention to me. And I mean that. And in some sense, I do mean that. I really am nobody. I'm just a guy on the internet. I am an undergraduate getting my bachelor's in field and organismal biology. And then afterwards, I'm going back for my master's in zoology and also a doctorate in zoology. So maybe one day I can be a real boy and do real research. But I have a YouTube channel, which is my name, Jackson Wheat. I do videos largely on evolution and debunking creationism. I also do interviews from time to time. So check me out if any of that interests you.
Awesome. Well, we appreciate you giving us your time. And yes, check out Jackson Wheat's channel. And thank you, Dr. Anderson, for giving us your time tonight. The floor is yours.
Yeah, my name's Dr. Kevin Anderson. I have a PhD in microbiology. I was a NIH postdoctoral fellow. I was a university professor for several years. Was the director of a tech company, currently working at the research center of the Creation Research Society.
Awesome. Well, thank you so much, Dr. Anderson. I'm not sure if it was just on my side. There was a little bit of breaking out there, but we'll make sure that there's no technical difficulties as we go on.
Well, the internet here is not the greatest, so yeah. The internet here is not the greatest, so it's possible that there'll be a little bit of breaking up.
Do we want to do my jetpack?
We can just work.
Yeah, no.
Let's
try it.
Okay,
Exactly.
we're gonna try to zip up our internet here.
Yeah, no problem. I mean, right now you're sounding great. If there's a little bit of breakup throughout the debate, that's fine. Well, okay, well, perfect. Before we go and hand it over to Jackson for his opening, though, I want to go over the format real quick for tonight. We have kind of a semi-formal, informal debate tonight, 10-minute openings, 5 minutes uninterrupted rebuttals, followed by a roughly 50-minute free-flowing organic dialogue. As always with these big debates, as moderators, we do our best to make sure that the dialogue is respectful and both debaters are getting as much time as they need to respond to each other's points. This will then be followed up by a question and answer period. And like I said, we've got a big audience tonight. So what we'll do is super chats will go first, followed by as many questions as we can get to. Please tag Erica, who in the chat will be Gutsick Gibbon with your questions. She'll save them, and we'll make sure we get to as many as we can during the Q&A. Anyways, enough for me. Let's get this going. Jackson, the floor is all yours. Whenever you're ready.
Can we wait just 1 minute?
Yeah, can you give us 1 minute to speed up our internet here?
So we might lose you for a second.
Yeah.
Of course.
Just give us 1 second. Sorry about that.
No, that's okay. That's okay. Take your time.
Yeah, taking time now and reducing internet problems on the line is absolutely preferable. Swapping out later. So
right. Let's get it all resolved now, and then we don't got to worry about it during the debate. So once again, like Gutsick Gibbon said, check out the after shows, Logical, Plausible, Probable, and on your channel as well, Gutsick Gibbon. So that should be fun.
yeah, it's going to be fun. I think it's a great opportunity to hear from everyone. It'll be neat. I'm excited to be here today. I've been looking forward to this all week. On a side note, I guess short week, but still.
No, me too. Yeah, this is ever since we set it up. I've been excited. Everybody has. We've had people camping out in the live chat all day, so this will be one to remember.
Yeah, I was also. I'm excited. This is this is the third. Anderson's the third person who has written for Answers in Genesis that I've spoken to. The first person was Mike Riddle, who RJ and I spoke to. I guess that was 2 years ago. It was a long time ago. And then the time flies, and you're having fun on the internet with randos. It's true. And then the other one was Dave Wotzel. I think that was actually 2017, and I haven't heard anything about his escapades in North Carolina trying to find the pterosaurs, but luck to him, I guess.
The ropin', yes. I think we discussed this the other day. The ropin'.
Yeah,
you can find those on your channel, Jackson.
Pterosaurs. I mean, I did do a video about dinosaur ancestors. So
Jackson,
So
I
they
think
get
you did the videos where you're talking to those guys.
Oh, yes. Also, you can find both of those on my channel way back. Well, I have I guess the 2017 playlist and the 2018 playlist is where you can check for those.
Whew, got them organized into the years. That's
Okay.
Everything is
We tried,
organized on my
but
channel.
It didn't work. So
Oh.
well, we'll work
All
with
right.
what we got.
We'll work with what we got. That's right. You sound good right now.
Okay.
Using the computer audio, sometimes if you're just talking as close to the computer and then the microphone as possible, there's usually not any issues. But if there is, we'll do what we can. So if
Okay.
everybody's good to go, if anyone has any questions, ask away. If not, we can just jump right into Jackson's opening.
Will there be snacks? Do we have snacks?
I know I've got snacks.
Oh well, I guess coronavirus, so we can't share snacks. Never mind. Sorry.
Not wise for the time being.
All right. Well, maybe next time. Okay. I guess whenever you want me to go, I'm ready.
Awesome. Yeah, whenever you're ready, go ahead. We can start the timer. And you got 10 minutes, Jackson.
1 sec. It's still opening. Okay. All right. We ready?
We are ready on my end.
Good to go.
All right. Here we go. Humorously, when Origin of Species was first released, Darwin had a relatively easy time convincing the scientific community that all of life was related. Rather, he had a more difficult time convincing them that natural selection was the mechanism behind biodiversity. Here, I'm also presenting the thesis that all life is related by natural common descent. Abiogenesis is entirely unrelated to this debate, as the last universal common ancestor likely lived many millions of years after. It also helps that creationists accept common descent to a limited degree. Creationists claim that the taxonomic rank known as a family is approximately the level of a baraman, which is an originally divinely created group of organisms whose descendants are all related by natural common descent. This, however, is problematic. For starters, all taxonomic ranks from genera to kingdom are arbitrary. There is no universal genetic or morphological metric by which species are delineated, hence the existence of species complexes and aggregates, nor genera, nor families, etc. These are merely man-made constructs that make the classification of organisms easier. A quick search of the literature on a genus with a long history, such as Nautilus or Lingula, will reveal numerous controversies over whether or not some fossil is a member of one genus or another, a testament to the fact that taxonomic categories are arbitrary and blur at the boundaries of branching lineages. What is important, though, is the fact that organisms are indeed related, and this can be shown a number of ways. For starters, genetic similarities imply relatedness. Paternity tests determine relatedness between a child and the father. A sort of extended paternity test can be done to determine the relatedness of all humans, and this test can also be done to show relatedness between species. Of course, creationists are fine with this for some species, but not others. Creationists are fine with using genetics and morphology to unite Panthera and Felis under the family Felidae, but not Pan and Homo under the family Hominidae. There is nothing intrinsically different about the process for either pair. The only issue is that creationists have a theological imperative to separate humans from the rest of creation. However, when we approach the data without such a bias, a different picture emerges. Remember that even Linnaeus, who was a creationist, grouped humans and chimps as the same genus. When we combine genetics and fossil data, we can see that all organisms on this planet are related by natural common descent. We can start with any species and move progressively outward until all of life is connected. For example, we did a recent video on my channel about hummingbird evolution. Two of the key differences between hummingbirds and their closest relatives, the swifts, is that hummingbirds have short wings and a modified elbow joint that allows for their hovering motion. Swifts, by contrast, have very long wings which they use for continuous gliding. However, there are fossil hummingbirds from the Old World, where no hummingbirds reside today, that have features in common with swifts. For example, Eurotrachilus has a coracoid like that of swifts, and Parorgornis has a swift-like beak. Further, Eosipselus, which is basal to both hummingbirds and swifts, has wings intermediate in morphology between both. This is precisely what we expect under common ancestry. Further, genetic evidence indicates that both hummingbirds and swifts are members of the clade Strasores, which also contains nightjars, oilbirds, patus, and frogmouths. And Strasores is a member of the clade Neoaves, which contains some 95% of bird diversity. Neoaves is sister to Galloancerae, which contains landfowl, ducks, and geese. Neoaves and Galloancerae form Neognathae, and Neognathae is sister to Paleognathae, which includes ostriches, rheas, emus, kiwis, cassowaries, and tinamou. There is no feature in any extant or extinct birds that indicates they are unrelated to each other. The same genetic studies that connect species within a genus also connect genera within a family, families within an order, etc. There is no clear break at any taxonomic level, which we should expect if different sets of birds are totally unrelated to each other. And morphological data indicates that birds are a subset of theropod dinosaurs. Birds have all the same bones as theropods, differing only in that some have been fused or are of a slightly different proportion in extant birds. For example, the ischium and pubis of modern birds are unfused at first during embryonic development, just like theropods, but become fused later. Birds also have prefrontal and postorbital bones around the eyes, just like theropods, that become fused and entirely undetectable during bird ontogeny. Recent paleogenomic studies have tracked some of these changes directly by experimentation, such as revealing the archosaur snout that became the bird beak. All the traits that birds have didn't appear instantly, but evolved gradually in their ancestral lineage, the dinosaurs. Genetics adds that the closest relatives of birds are crocodilians, which is extremely bizarre, until one looks at the fossils connecting the two. Dinosaurs and birds have an advanced mesotarsal ankle joint, while crocodilians and their extinct kin have a cruratarsal ankle joint. However, all archosaurs have antorbital and mandibular fenestrae, as well as a pronounced fourth trochanter. Archosaurs are a subset of diapsids, which are sauropsids with two temporal fenestrae. All tetrapods that develop embryonically inside amniotic eggs are more closely related genetically to each other than to anything else. Sister to sauropsids are the synapsids, the mammals and their ancestors. We have a near perfect fossil record of the jaw to ear bone transition in mammalian ancestors, including predicted fossil forms like Probainognathus. And we recapitulate the movement of bones from our jaw to our middle ear during ontogeny. Of course, genetics shows that all tetrapods with 3 middle ear bones, lactomammaries, and hair are more closely related to each other than to anything else. And creationists, in general, try to avoid the subject and don't discuss most of it. All vertebrates with 4 limbs are more closely related to each other than to anything else. We have a very nice transitional sequence of tetrapods developing from earlier Sarcopterygian fish. All vertebrates are more closely related genetically to each other than to anything else. We have a fossil record of early vertebrates and chordates, such as Hycoichthys and Metaspergina. On the protostome side of Bilateria, we have a beautiful fossil record of early arthropod evolution, such as the various lobopodians, as well as fossils documenting the ancestors of mollusks, such as Halkieria. The recent discovery, Icaria wariutia, may be representative of ancestral bilaterians. All bilaterally symmetrical animals are related genetically and morphologically, and bilaterians, cnidarians, placozoans, ctenophores, and porphyrians share genetic similarities, as well as being eukaryotic, multicellular, and phagotrophic. The closest relatives of animals are protistine choanoflagellates, filasterians, and ichthyosporians, all of whom share a fusion between the gene for ubiquitin and the gene for ribosomal protein S30. What an oddly specific fusion to have in all of us if we're not related. Further, animals and these protists, as well as fungi, apusozoans, and amoebozoans, share a gene that is a fusion of 3 genes for enzymes involved in pyrimidine biosynthesis. Another curious detail. All of the aforementioned clades plus plants, diatoms, forams, coccoliths, brown algae, and euglenozoans, among others, are related in that they all have cells with a nucleus, mitochondria, and various organelles. They are eukaryotes. There was also an extraordinary fossil record for the tough shelly protists, which make them great index fossils. Certain trilobites, conodonts, graptolites, bivalves, ammonoids, and other organisms are also index fossils. That is, a particular species exists very briefly in the fossil record, but is able to be correlated with a specific time period. It goes without saying that those sorts of fossils shouldn't exist if a global flood happened and the Earth were only 10,000 years old. And once again, creationists rarely address the distribution of these index fossil organisms. Finally, all of life is related. all organisms have DNA, RNA, and make ribosomes, as well as require water, metabolites, and space. There is no reason for all organisms to have these features unless they had a common ancestor who also did. Bear in mind that if a single all-powerful designer created all life, then this nested hierarchical pattern shouldn't necessarily exist. An all-powerful designer could, by definition, create anything. It could create all Barrowmans as totally separate from each other, and really shouldn't we expect that? Shouldn't there be some easy divide that separates every Barrowman from everything else? The best evidence of a designer would be for that designer to give every Barrowman a different type of genetic material, drawing on codon assignments unique to each, or for the designer to make genes and morphological characters in such a way that phylogenies only return polytomies. Was the designer not clever enough to outwit 21st century cladistics? There is, by contrast, only one pattern that evolution could create, a nested hierarchy. The reason for this is that evolution is descent with inherent genetic modification. It must modify that which already exists. To conclude, all of life fits in a nested hierarchy, which is the sole path that evolution can take. If you want to argue that a designer would necessarily create this pattern too, then that designer is indistinguishable from natural evolution and is thus not necessary to postulate an explanation. There is no single useful metric by which to delineate Barrowmans, and all of life is indeed related. Thank you.
Awesome. Thanks so much, Jackson. Just on time, actually, with a couple minutes to spare, roughly. So we can toss that into some Q&A, which everybody looks forward to. Yeah, thanks so much for the visuals. Clear, concise, great opening. Dr. Anderson, whenever you are ready, take your time when you're comfortable. We can get, if you have slides, we can get that shared for you as well. And I'll put 10 minutes on the clock.
Can you still hear us, Dr. Anderson? Just to make sure.
He may just be muted from Jackson's opening.
Yeah, maybe.
Yeah, that might be that might.
There we go. Doing the screen share.
Sorry,
There we go. No
we
problem.
couldn't. It was muted and it wouldn't let us unmute.
No problem.
No problem.
Okay. All right.
There we go.
Okay.
Yep.
Gotcha.
We'll start the
Okay.
timer on your first word.
All right. All right. I cannot necessarily say that Charles Darwin was the first to propose that all life on Earth shares a common ancestry, but it was a core principle in his Origin of Species. He frequently alluded to descent of life, and he first sketched his concept of a common ancestry tree in 1837. While Darwin spent most of his writings discussing small phenotypic changes in different pigeons and dog breeds, he assumed that these little changes would continue to virtually unlimited. In fact, this unlimited number of changes was a major assumption in all his writings. Enough little changes gradually adding up to major changes, so ultimately invertebrates would evolve into vertebrates, fish evolve legs, reptiles evolve wings and feathers, and on and on. With the possible exception of an Austrian monk, are we frozen up?
Something's happening. Just keep going.
Well,
Keep going.
but it's
We can still see your screen. I'm not sure.
We can still, and if there is an issue there with the changing slide, we can pause the timer too. So
Yeah,
don't worry.
I got a timer paused right now as well.
okay. We got it. Just sorry. Are we before? Oh, all right. Go one more. Okay.
Go.
All right. Jeez. Having fun. Okay. With the possible exception of an Austrian monk, no one in the 19th century understood anything about inheritance or how organisms change. Darwin proposed that natural selection was magically create traits each organism needed for improved fitness and increased survival. And some version of this remained in the evolutionary literature for decades. In fact, it still seems to subtly be there even today. However, structures of DNA were not resolved until 1953. 3 in a basic understanding of genetics plus years and was very little idea of how genetic information worked. As a consequence, the universal evolutionary ancestry of all life became entrenched in scientific thinking without any understanding of a mechanism other than the non-existent creative power of natural selection. Yet such understanding is vital to any process of universal common descent. More recently, mutations have been added to the mechanism, and these are now deemed as the driving engine of evolutionary change, and hence the driving engine of universal common descent. Today, mutations plus natural selection is the central axiom of universal common descent. By randomly changing an organism's DNA sequence, mutations are assumed to provide the biological world a continual source of new and untried DNA sequences, perhaps analogous to new books suddenly appearing in a library. Natural selection then sorts through all these new sequences, discarding the bad, ignoring the inconsequential, and finding the ones that help the organism run faster, fly higher, see further, or whatever beneficial trait results from the mutation. However, natural selection is conservative. It cannot create a trait, as Darwin apparently presumed. It can only eliminate traits from a population. In fact, its elimination power is what Fisher's theorem of natural selection promoted. For decades, Fisher's work has been heralded as a mathematical proof of how natural selection drives a population to increased fitness. Yet, Fisher assumed that the number of deleterious and beneficial variants would occur with equal frequency in a population. This is demonstrably false. Thereby, Fisher's supposed proof of evolution is false as well. Even Kamara's seminal work on population genetics, published in 1979, recognizes that virtually all mutations are either neutral or deleterious, and that natural selection cannot see and rid a population of all these mutations. It is simply too nearsighted. While epistasis and forms of hard selection are offered to increase natural selection's down of all populations. This, of course, contradicts the claim that life on the planet has been evolving upward and onward for the past half billion years. The very process that supposedly established the massive transformations on Earth past would have actually driven all life to extinction long before now. Nor can mutations be viewed as a sufficient source of raw genetic material for the massive transformations envisioned by universal common descent. With few exceptions, even mutations clearly providing some benefit to the population, as transient as that benefit often is, are still degenerative. Antibiotic resistance, lactose tolerance, HIV resistance, DDT resistance, increased plant growth rates, and numerous other examples of evolution in action result from mutations that reduce or eliminate pre-existing genetic activity. Even the famed citrate mutant of Linsky's long-term evolution experiment was little more than a rearrangement of a pre-existing gene to a pre-existing regulatory system. No new genes or regulatory systems were formed. What is more, the citrate mutant is now dying from the weight of its own mutation overload. Further evidence that of the inability of mutations plus natural selection to move any population too far from their starting point before the inevitable death spiral as mutations accumulate. A long-term evolution experiment with fruit flies had a similar lack of success. After 600 generations, no new unconditional beneficial mutations became fixed in the testations. While the spontaneous formation of new genes is assumed by universal common descent, the sheer improbability of this is daunting. In 1966, a group of evolutionary biologists and mathematicians gathered at the Y-Star Institute in Philadelphia. In short, the mathematicians showed the extreme improbability of randomly generating the necessary mutations in one gene, let alone numerous genes, to spontaneously appear. What is the mathematicians? The biologists simply argue that evolution happens, so your math is wrong. In other words, their supposition that universal common descent has occurred skews their interpretation of everything else. To them, universal common descent is not a theory to be tested, but rather an axiom used to interpret and test all other theories. While the fossil record is often claimed to show evolutionary common descent. This is only a historical reconstruction. In other words, at best, it provides only indirect evidence. Yet even within this reconstruction, there are numerous problems. Number one, major taxa appear abruptly in the fossil record with little evidence of their ancestral history. As justification, I have heard evolutionists argue that we do not need to see grandma at every mile marker in her train trip to know that she successfully made the trip once she exits the train. So it is with evolution. We do not need complete fossil documentation of the intermediate stages throughout time to see that organisms today have evolved from ancestral organisms over vast periods of time. However, by this analogy, the evolutionists are expecting us to accept on faith that when grandma started, she was a short, gray-haired lady of medium build. But by the time she exited the train, she was a tall blonde of slender build. They should not be surprised that we asked for more documentation of the transformation during this trip. So it is also that if evolutionists expect me to accept a fish as transformed into an amphibian with an entirely different anatomy and physiology, I need more documentation than one or two highly arguable and not very revealing fossils. Number two, universal common descent is void of an adequate mechanism. Mutation and natural selection are simply not up to the task. Yet without an adequate mechanism, any historical reconstruction lacks explanatory power and actually then is not to be viewed as a correct reconstruction. Universal common descent is strictly a presupposition imposed onto the genetic data, not a conclusion drawn from the genetic data. Remember, universal common descent was popularly accepted long before anyone knew anything about tRNA, microRNA, promoter sites, and other basic genetic systems, let alone the elaborate and extraordinarily advanced genetic systems that are now understood to exist, such as epigenetics, a level of genetic information layered on top of the actual DNA sequence. Number three, it is not just the assumption of random mutations that fails. Recent work challenges the time frame which beneficial mutations can become established. Called the waiting time problem, it is calculated that it takes several million years just to fix a few beneficial mutations into a population. Universal common descent cannot function within this genetic restriction. Number four, pseudogenes and other supposed junk is not evidence for common ancestry. Initially, it was assumed pseudogenes were broken genes, and if two organisms, for example, human and chimp, shared the same broken gene, they acquired that broken gene from their common ancestor. Yet, as more and more pseudogenes are being found to serve very important functions, along with other so-called junk DNA, there's little reason to assume these represent broken genes. If they are not broken genes, then they do not provide evidence of an ancestry. This argument is simply an outdated evolutionary extrapolation. Number five, fossils can be arranged in a variety of ways depending on the desired lineage. If all we knew of dog breeds were some fossil fragments, it would be very easy to impose a lineage where none actually exist. The same can be said for alligators. Even though these are all living alligators, arrange them to show evolution of small to large or large to small, depending on what direction you're wanting it to go. Ultimately, universal common descent was first proposed with no evidence, only a presupposition that goes on to the interpretation. Now, decades and volumes of research data later, it still is only a presupposition imposed onto the interpretation of the data.
All right, thank you for that. Standing, if you want to take over from here, I just no dead air alive. Okay, keep it as an
Yeah, no, awesome. Yeah, nice opening from the both of you. I appreciate the visuals. I know it takes a lot of work putting together those presentations. So good job to the both of you. We would now be moving into the 5-minute rebuttal, which as moderators, we will have to be a little bit more strict with. So the second you guys start to rebut the points, we'll start the clock at 5 minutes. Jackson, whenever you're ready, we can start the clock.
We can certainly, with pleasure, do that.
I believe it because one time it was a debate we had recently where with the webcam there was some issues and turning off the webcam seemed to resolve those. So it's worth a shot. That being said, thanks for the suggestion. Praise. Jackson, whenever you're ready.
Okay, I probably won't take the whole 8 minutes. I made a couple of notes on here. He mentioned Kimura's work. Well, he mentioned a lot of things. He mentioned Kimura's work, and it says that most of the mutations are neutral harmful. Yes, that's true, but even as Kimura pointed out in his famous 1974 paper, the beneficial mutations will vastly outweigh the neutral and harmful ones in terms of fitness. In fact, I know John Sanford's entire model is built off of his misinterpretation of Kimura's 1974 paper because he thought that, or he seems to think that Kimura was saying they don't happen or that they're unselectable, when in fact he was saying the exact opposite, that beneficial mutations do occur and are highly, highly selectable, vastly outweighing neutral and harmful ones in terms of fitness. He mentioned lactase persistence. Thanks to Dan Cardinelli, actually, for preparing me for that one in advance, because I know that the argument you use for that one is that the MCM6 gene control, which it controls a repressor or codes for repressor, and then that got broken. However, that's not at all the case. In fact, it's this gene does not code for repressor; it codes for a helicase. So it's not at all what you claim or have claimed before. So there was nothing broken there. Another example with the E. coli. So with the E. coli, there nothing was broken there. You're right. There was a movement around. Was it? It was into. Nothing was. What did I say? What did I say? Lactase. Yes, MCM6. Yeah. Sorry. It blocks normally, but a mutation prevents repression. But it doesn't make a repressor codes for part of a helicase. I said that. Okay. Yeah, I mentioned the E. coli. So there was a duplication for a promoter region, and the promoter region got moved. There was nothing lost there either. I don't really understand what the problem with that is. You mentioned that's what happens in evolution. There was a paper I looked at recently that was talking about phylogenomic comparisons among the kingdom Animalia and their closest protist relatives. And what it appears in most lineages is rather than a whole bunch of new genes coming out of nowhere and coding for new things, rather genes that already existed were duplicated and neo-functionalized in a bunch of different lineages, and this instead is what produces new organs and things like that. In fact, there was a recent example with Procambarus virginalis, which resulted from a speciation event that was caused by a whole genome duplication event. So there's a good recent example of that. The 1960s symposium, Stephen Meyer makes this argument too, saying that the mathematics shows that evolution can't work. I can't seem to get to the actual symposium itself, but I have read reviews of it by a biologist, and rather it seems that the symposium was really about mathematical challenges to neo-Darwinism, was really about being able to make mathematical models which accurately model neo-Darwinian evolution, which is very old now. The modern synthesis is not neo-Darwinism. It's not simply mutations plus natural selection. It's nothing so simple anymore. What I remember from the paper that I read on it was that, that the biologists weren't really convinced by it, but that there were mathematical models that could work for it. And as time has gone on, we have computers now and all sorts of things, and we can model evolution very well. You mentioned pseudogenes. All right. Gulo, the pseudogene for L-gulonolactone oxidase, is my favorite pseudogene because it's both an example of common ancestry and also a good example of something that doesn't work. It doesn't work, and we know it doesn't work because the promoter region is broken. There's nothing for it can't be transcribed. RNA polymerase can't transcribe anything because it can't bind to the promoter region.
10 seconds.
Whoops, I guess I did cover pretty much everything. Not all antibiotic resistances, methods of antibiotic resistance are the result of loss. Sometimes they find efflux pumps and other fermented mechanisms. Sometimes it's carried on plasmids, which are transferred by horizontal gene transfer. So no, it's not always a loss. So anyway,
All right. Thanks for that. We'll jump right into Kevin Anderson's, Dr. Anderson's, 5-minute rebuttal. Standing, do you have like any questions about the format? Because we were just talking about that in the side chat.
No, it looks like Jackson went for 5 minutes, so we will do, I guess we got a little confused there if it was 5 or 8, but Jackson went for 5, so we will go 5 minutes for Dr. Anderson as well.
All right. We'll begin the time on your first word, Dr. Anderson, and don't forget to unmute yourself.
You're
You're
unmuted.
Okay. All right. Very good. All right. Well, first off, to go to Linsky's long-term evolution experiment, a lot of the mutants that he obtained were losses, which makes sense if the organism can get rid of stuff and actually replicate faster. So that would be clear predictability, but certainly not what's going to drive universal common descent. My comment specifically was on the citrate mutant, which I didn't say was a loss, it was a rearrangement. But see, the key thing I said was that there was no new genes or regulatory systems formed. It was rearrangement using pre-existing genes and regulatory systems. And universal common descent isn't going to get very far if it simply just uses what's already there. You've got to start being innovative and coming up with new sequences and developing new stuff, because you're not going to get from a lizard to a bird by not providing something new in the genetic structure. So I just simply use the E. coli as an example of that they didn't really get what they were wanting to herald that they were getting. And in fact, now that the mutant strains are actually beginning to die because they have such a heavy mutation load. And I use as example that natural selection plus mutation is not able to keep it clean and organisms will die. You commented that John Sanford misinterpreted Chimera's work. I would strongly disagree with that. And in fact, Lynch and Crow and others along the lines that also do population genetics would agree with John Sanford that they are struggling to figure out how in the world things have not decayed away. They have all kinds of explanations. There's a book that Klazonsky wrote that's The Crumbling Genome, where he spends a lot of it trying to figure out how can we explain that the genome is still there and hasn't essentially mutated away. And of course, junk DNA is one of his favorite explanations, just like it's one of Gar's favorite explanations too. And so they need junk DNA in order to be able to, if you will, hide all the mutations that occur. That's why if junk DNA starts disappearing from the scene, then they're in big trouble, because now they can't hide the mutations anymore. I found it interesting also, yes, you are correct in antibiotic resistance. Not all resistances are mutants. There are some that come as a result of horizontal gene transfer, and that is very true, but horizontal gene transfer doesn't explain its origin. It just simply explains how that particular genetic system, that particular genetic gene series of genes is moved around within a population. It doesn't explain where it came from to begin with. And I find it also interesting that the genetics do not fit very well universal common descent, which is why Doodle and others have had to go to this meshed up, scrambled up tree to try to rely on horizontal gene transfer to explain why genes suddenly appear and disappear all throughout. And that is not consistent with these nice lineages. Plus, they're taking a lot for granted with horizontal gene transfer that has never been shown to actually occur. They're just, again, imposing a reconstruction onto it that, oh, this must have come from horizontal gene transfer, because where else would it have come from? How much time do I have left?
Yeah. 2 minutes.
2 minutes. Okay. The idea that one of the evidences for common descent is that we all share the same DNA, RNA, all breathe the same oxygen. You know, we live on the same planet as all other life on this planet, so it shouldn't be surprising at all that all life on this planet would share certain similarities because we live in the same planet, breathe the same oxygen, have the same availability of the different molecules of proteins and carbohydrates and lipids and such. So I don't really see how similarity of RNA and DNA somehow means that universal common descent must have been the explanation for it. Let's see. Again, it comes back to you correctly made the comment that species and genus and family and such are man-made constructs. And because of that, there's a certain fluidity to it. And so it is hard to necessarily know where to draw the lines, which is also interesting why in the fossil record you see such stasis, even when it's difficult to draw the lines.
30 seconds.
Oh, I got 30 seconds left. Oh, okay. It still comes back to that universal common descent lacks a genetic mechanism. And as a geneticist, to me, if you have no genetic mechanism, then it's just a reconstruction imposed on the data. If you cannot explain how it happened, then don't tell me it happened. Okay, so I guess my time's up.
Yeah.
Perfect timing. Great rebuttals from the both of you. And actually, Dr. Anderson, your audio was great there. I didn't hear any issues. I'm not sure if anyone else did.
Okay.
So I think turning off the webcam and just kind of saving some bandwidth worked. So that looks good. Yeah, so now we're jumping into the open dialogue. As I said in the beginning, you know, we're going to keep it civil and fair in that both debaters should feel they are getting equal time to address each other's points uninterrupted. As moderators, you know, we like to sit back and kind of keep it an organic, free-flowing dialogue, but we will intervene if necessary. I don't think it'll be necessary. You guys are both respectful, but if we have to, we will. So I guess since Dr. Anderson finished his rebuttal, we can start with Jackson Wheat with a point if you'd like to make, Jackson, or a question you'd like to start it off with.
Okay, so no, Kimura's work is most certainly misrepresented by Sanford. Kimura argues that, yes, neutral and deleterious mutations are the largest percentage of mutations that occur. However, which is why he didn't include, or the reason he didn't include beneficial mutations in his analysis is that, as he pointed out later in the paper, these vastly outweigh, though they're far fewer, they vastly outweigh both neutral and harmful mutations in terms of effect on the organism. Sanford claims the exact opposite, that they basically don't occur. I don't understand how that could possibly be, how that's not a misrepresentation of his work. It's like saying
John is not saying that they don't occur. He's just saying that they occur very infrequently and they are very easily lost among all the other gamish that occurs within population genetics. And in fact, regardless of your interpretation of Chimera, the genetic evidence, the experiments, such as the Drosophila experiment I mentioned in my initial opening, they couldn't even, after 600 generations, they couldn't even get a single unconditional beneficial mutation to be fixed in the population. So that's totally contradictory to what you're saying Chimera was arguing, because you're saying by his argument they should have had no problem at all. They couldn't get a single one to be fixed. And we see this repeatedly in the genetic experiments that are done where they try to fix in populations beneficial mutations. It just is not very easy to do. And often what happens is you don't get them fixed at all.
It may not be easy to do. I didn't say it necessarily was. In the example of the E. coli, the citrate mutation did become fixed in the population, though.
It became fixed in the population, and now it's dying.
According to who? Do you have?
According to, I gave the paper in my talk. It's Linsky's latest paper in eLife 2020. The clones are dying.
All of them are dying.
Well, I'm not saying all of them are dying, but a lot of the clones are dying. Because remember, all the clones he obtained, they all have different mutational loads.
Do you have the paper on hand, or do you know what the title of it is?
I do. Here you go. It is eLife 2020 Volume 9. And then, of course, because it's an online journal, it has the weird page number. So it's E55414.
Oh, what's the title of it?
Genomic and Phenotypic Evolution of E. coli in a Novel Citrate-Only Resource Environment.
Okay, I found it.
Unexpectedly, some evolved populations and clones show apparent declines in fitness. We also found evidence of substantial cell death in citrate plus clones.
And
that's what happened.
It looks like they added it. Okay. We previously reported that aerobic growth on citrate evolved in a population of Escherichia coli during adaptation to a minimal glucose medium containing citrate, which is DM25. CIT plus, which is the mutated one, can grow in citrate only medium, DM0, which is a novel environment for E. coli. To study adaptation to this new niche, we evolved one set of CIT plus populations for 2,500 generations in DM0 and a control set in DM25. We identified numerous parallel mutations, many mediated by transposable elements. Several lineages evolved multi-copy amplifications containing the MAA gene, constituting up to approximately 15% of the genome. We also found substantial cell death in ancestral and evolved clones. Our results demonstrate the importance of copy number variation and transposable elements in the refinement of the CIT plus trait. However, the observed mortality suggests a persistent evolutionary mismatch between E. coli physiology and a citrate-only environment. That's interesting. That is interesting. So,
and that's back to my claim that mutation plus natural selection only can get you so far. Yes, you can get variation, as you correctly pointed out. Even the creation model will allow for variation. It's how much variation is acceptable. And my challenge is that when you're talking about the massive amount of variation they're going to be involved in going from, a lizard to a bird or going from a fish to an amphibian, that's where you run up against that you cannot accommodate the mutational load that's going to occur, and natural selection just simply can't keep it clean.
So in essence, they moved it to I don't know where that came from. So they moved it to a new environment, and it's not as fit as the original population was. I don't find that especially compelling. I mean, this is an interesting study, and I will read it I'll read it later tonight, but I'm not hugely surprised that this occurred. So, okay. I also don't think that this somehow means that mutations are not the mechanism, or they're not providing for the raw substrate for natural selection to act on. It seems that natural selection is indeed occurring. So I'm
not challenging that. I'm not challenging that mutation occur. I'm not challenging that there is a selectivity that occurs. I'm challenging the level of mutation and the level of selection necessary to go from Luca to human.
And this doesn't really seem to demonstrate that.
No, its demonstration is, of course, as I pointed out, its demonstration is, first off, after all was said and done, nothing new had evolved as far as genes or regulatory systems, just the rearrangement and adaptation of pre-existing systems. And after enough time, the mutation load comes so much they start collapsing.
Well, that's not what this is saying either. It looks like it's saying
that is what it's saying. That is what it's saying, is that they have a high rate of death in the citrate mutants now.
Between the E. coli physiology and a citrate-only environment. This isn't saying, as far as what I can see, just because I can only see the abstract here, I'm trying to open it up.
Understand.
Trying to open up the full paper, but it just seems from this that it's only saying they changed the environment. But like I said, I'll try to read this later. I'll try to read this afterwards so that I can get the full picture, but it doesn't seem to be saying what you're saying it says. But like I said, I'll read more of it later. But regardless, what happened with the formation of the citrate of the citrate plus lineage is this is a novel thing that happened. They're able to metabolize citrate aerobically. That didn't exist in their population originally, right? There was a duplication of the promoter, and then it moved. It was translocated to a new region where it was put in control of the of citrate metabolism, but now it can do it in an aerobic environment, right? So that is a new trait. They couldn't do that originally, but they can do it now, right?
Right. Not going to say it's not a new trait. Again, traits come and go, and sometimes traits related to genotype, sometimes they're not related to genotype.
Sure.
But the point still comes back to get this trait involved using pre-existing gene and regulatory system. The trait did not come about because a new gene or new regulatory system evolved.
Well, this is a new. Well, I mean, it's not a totally new one, but what existed was changed, and that caused a new function to occur, right? And as I mentioned,
but by moving a pre-existing gene next to a pre-existing promoter, that doesn't explain the origin of either the gene or the promoter. It just explains what you typically get in bacteria, and those bacteria are constantly rearranging their chromosome all the time. I would suspect that's to make up for being asexual organisms, but that's just kind of my idea. They have IS elements that move in and out all the time to try to generate genetic diversity. But again, it doesn't explain the origin of the IS element, the origin of the regulatory system, the origin of the gene. It's just simply what's already there, and you're rearranging stuff to get new traits.
Okay, but couldn't you say that for like feather formation, for instance, since the genes that code for the keratin that makes up feathers are duplications of the genes that code for the keratin that makes up scales? So the entire thing is just a series of duplications within duplications. So would you say then that feathers aren't a new structure, even though they evolved by duplications within duplications?
It's not surprising that there would be some similarity since they both involve keratin. But it's not that simple, because a scale is structurally much different than a feather, and embryological control, which involves the Hox genes and everything else, you're going to see a lot more elaborate activity going on to form, or a lot different activity going on to form a feather versus a scale. So yeah, it's not surprising there'd be some similarity because they're using the same basic protein. But the event of making the scale and the event of making the feather involve totally different regulatory and protein systems.
So actually, the gene scale, which is involved in the keratin that makes up scales, hence its name, is also the gene that is involved in the makeup of feathers, or the homolog of it is involved in the gene that makes up, or that codes for the.
Okay, you'll have to define when you say make up. It's
regulatory. It's regulating the process by which feathers are made.
Okay, so it would be one of the regulatory systems involved.
Yes, hence why I'm saying that it's involved in the process of making scales, and yet it's duplicated, and there are new gene families that are birthed from that duplication event, which are involved in the process that regulates the development of scales.
That to me, that's imposing common descent on. You said there's new gene families. So evolution just came up with new gene families. They just happen to be there, so they must have evolved. My challenge is that they didn't evolve; they're already there. So because
sorry, go ahead.
No, that's fine. That's fine. I didn't mean to interrupt you.
No. I apologize.
I was just going to say that when you have a series of genes, and some are shared and some are not, universal common descent assumes that the ones that are not have subsequently evolved and subsequently formed. And my challenge is there's not a mechanism that's going to do that. We don't have a mechanism because mutation does not do that. It's never been ever shown to do that. It's just assumed to do that. When you talk about orphan genes, well, orphan genes are assumed to just be de novo genes. But that's just a historical reconstruction.
So
Because you cannot go into the laboratory and demonstrate any series of mutations that will give you an orphan gene from what they're saying it started with. Most orphan genes require at least 5 or 6, if not 7 or 8 mutational changes in the same gene. And you can't go in the laboratory and do that. So to me, that's just a historical reconstruction. We're just assuming that they de novo.
So you would
I didn't
say
mean to take up all your time.
oh no, you're fine. I think this broaches a good topic. So oftentimes orphan genes are restricted to species, right? That's largely. Although they occur at all sorts of different levels in taxonomy, right?
Yes, they do.
Okay,
right.
So they occur largely at species. So if that's the case, are you saying that you also agree that de novo genes occur naturally, or is every species have de novo genes that were just given to it divinely?
No, the most likely explanation from a creation model would be that speciation is a process of diversifying the initial gene pool. And so one of the species got a certain amount of the gene pool, which had, let's say, orphan gene A, B, and C, and another species got other part of the gene pool, which had orphan gene A and B, but not C. And it's not a matter of that during the diversification, you had a de novo formation of the gene. It's just how the speciation separation of the gene pool occurred that gave you the speciation to begin with.
So that would require vertebrates with ridiculously high amounts of chromosomes? How would that work? How is it possible to construct a family tree by which only certain sets of these portions of the total variation within this population of organisms were split in certain ways when it's coming from the same ancestor?
How far back are you going?
If
I'm
we're
probably
looking,
not going near as far back as you're going.
I mean, what you're proposing would come up with highly non-parsimonious phylogenies, because you're talking about these things are lost in all of the other relatives of this thing except for this one, which is extremely non-parsimonious. If we're looking at like different species within a genus, and each species has its own de novo gene, if each species has only one de novo gene, how is it possible that the ancestor of this cluster of species had all of these de novo genes, and yet none of these de novo genes show up in the other species? They're only in each one. How is that possible? How is it possible to construct a phylogeny like that? Because that implies that the ancestor had either lots of chromosomes or this just doesn't make any sense.
It's much easier to destroy than to build. So it's much more likely that some of the contemporary organisms, the orphan gene has been lost in them from mutation than it is that there was a de novo formation of the orphan gene.
So again, I have to ask, how is it possible that they just that you get these sort of perfect distributions where where these de novo genes aren't ending up in anywhere else, either by recombination, for instance? They haven't ended up in any other lineages. They've only ended up in certain lineages. It certainly looks like natural evolution, like these de novo genes are evolving in different lineages. Why is it that it only looks like that?
Well, I'm not saying it looks like that myself, because I don't see the mechanism that you get de novo genes. So, and it's amused me because 20 years ago, evolutionists were very willing to admit that genes are going to take a while to form because it becomes ridiculously improbable to be able to get the right 5-6 mutations within a sequence of DNA to form a gene. And now all of a sudden they have all the orphan genes. It's like, oh, de novo genes just happen all over the place. And it's very interesting, in 20 years, they've totally switched their tune. Not the genetics hasn't changed, just the need for evolution has changed, because now they got to explain orphan genes by de novo genes, not by degradation or deterioration.
You're still getting us to ridiculously non-parsimonious phylogenies, and I think that really needs to be addressed, because I don't understand how it's possible that none of these other ones got the recombinate or through recombination ended up with the de novo genes. It's just these particular species. That does look like natural evolution. The fact that a certain set of organisms all have this one de novo gene, and I think you would agree that two species within the same genus are related to each other, right? Unless you wouldn't.
Depends, especially in the insect world, where a lot of the orphan genes have been studied. The insect world is just bizarre. And so I try, when it comes to answering that question in the insect world, I tend to back off because I don't know, because the insect world genetics is kind of strange. So I cannot answer your question because I'm not sure how much I'm going to go back to the original creation. So, because insect genetics is just strange.
Okay, so we'll take something like birds, for instance. Like, if we're looking at like tanagers, like from the Galapagos Islands, like Geospiza magnorostris and conorostris, you would say these two species within Geospiza are related to each other, right?
Well, I'm not an ornithologist, so I can't necessarily say I would or not. We'll just, for the sake of the argument, okay, I'll say yes, just for the sake of the argument.
Okay, so these two are related to each other, right? And each one has its own orphan genes, but they're related to each other. And there are other members of Geospiza, which I think you would also agree are probably also related to them. They're all in the same family, which is Theropidae. But they're probably all related to each other, right? I mean, even if you
again,
for the sake of argument,
for the sake of argument, I'll say yes. I mean, because I'm not familiar enough with those species to be able to tell you where I'm willing to draw the line. Of course, that's the problem with birds, as we find a lot in biology. You have species A, species B can interbreed. Okay, so is species A and species B the same? All right, well, but species B and species C can interbreed, so are they the same? But then species A and species C cannot interbreed. So where's the line at?
Yeah, I absolutely.
That's part of what, right? Yeah, that's part of what happens when you get diversification going on. And I don't know if anybody, evolutionists, creationists, can explain all of that, simply because there's a lot of genetics that's not understandable yet when you have these kind of lineages going on.
And yet in each of these groups where they are related to each other, for whatever group that you agree is related, to or whatever group you agree where all the members are related to each other, they're going to have orphan genes, probably within the species that's in one species but not in the other species. But from my estimation, that would be because after the species split, they develop their own. But again, orphan genes occur at like all taxonomic levels. There's an orphan gene for for the genus, for the total genus, and they're all related to each other. There's a there are orphan genes within the species and higher than that. But I would say that because they're increasing sets. This is the nested hierarchical thing, which is also something I do really want to broach with you. The topic of horizontal gene transfer and Doolittle at some point. But I did write it down. But still, it's. Again, you're asking for very, very non-parsimonious phylogenies to be made. If these things are all related, it just so happens that these de novo genes appeared in were selectively lost in every other lineage except one. Every other lineage leading to these modern species except one, and that I don't. There's no evidence for that. That would be.
But how much does it take to lose an orphan gene?
That's a good question. How much does it take? Why would it be lost? If we're talking about variations, why would it be lost in all of them except for this one? And why is it that why are these groups related but not to this other group which also has its own orphan genes? Why is it that it was lost in some of these lineages but not others? Why do some share these certain orphan genes but others that are also within this group don't share these orphan genes? What is the reason for that?
And why is it that primates have more chromosomes than humans if you're saying that they're descended from the same ancestry?
Oh, that was a fusion event between chromosomes.
Well, yeah, that fusion event, that's pretty bogus. That was
It's really
jumped on because it was what was necessary. But if you go in and you look at the sequence, you don't have the necessary sequences there of the centromere or any other evidence of telomeres or anything to suggest that that area they're saying was fused was actually two separate chromosomes.
So actually you do have both you have a degraded telomeric region, and right next to it you have a gene family which is a telomere specific pseudogene family in the middle of the chromosome arm, directly adjacent to the region that is supposed to be the alleged telomere region. Now, if that's not evidence for a telomere, I don't know what the evidence would look like.
I'm going to totally disagree with you there. You have no more trace of telomere in that area than you do. Anytime you sequence, you'll find traces of telomere sequences throughout the chromosome. There's no more telomere sequence in the supposed fusion region than there is throughout the rest of the chromosome, except the telomere site. The telomere
actually, there is. Because, and here I can demonstrate it. So the region, the telomere sequence, what is the sequence? Is it CCA TTG? What was the sequence? I don't remember. But anyways, you're right. That sequence on just at random is found in other places. What it isn't found is 125 times anywhere else in the genome except that one spot. There's only one spot in the entire genome where it's found, and it's that one, which is the spot right next to the telomere-specific pseudogene. So again, if that's not evidence,
you will have to give me the paper for that one because I have never seen that.
Oh yeah,
I've never seen anybody report that.
Dubs Ruhif has done a bunch of stuff on. Actually, let me save this one, which is the paper you sent me, so I can look at that later. Let me save that. PZ Myers did a video on it way back.
Really quickly.
All right, I don't count PZ as an authority on anything, so move on.
Okay. All right.
I hate to interrupt here, but I think that after we settle this question here, I think Dr. Anderson should ask Jackson a question. You should get time to ask Jackson a question.
Go ahead. You can go ahead.
So I've been asking him questions. That's fine. That's fine. I'll ask one question when we're done. But it's been a fair exchange. So I've been asking him questions back. So I'll do one after this.
Sure. Do you have actually? Well, let's see. Let me see if I can.
I would like to, because I've never seen a paper reporting 125 times the telomere sequence in that site. I would like to see the paper on that.
All right, let me see.
In the meantime, Dr. Anderson, if you would like to go ahead and ask your question, maybe Jackson can.
Apologies, I'm looking for his blog right now. I'm sorry.
I'm asking him to look it up. So
yeah, no problem. I want to make sure y'all you guys feel like the conversation is moving right along. So it's up to.
I'm having a fun time.
Yeah, it's a discussion for sure.
Everybody in the live chat's having a great time too. This is very engaging. Keep it up, guys. Very, very good dialogue.
We have a lot of questions for the Q&A for the both of you.
Okay, let me see. Here's his YouTube page. Let me see. Does he link to his? Good Lord! If someone in either the live chat could link to the blog for Glenn, what's it, Glenn Williamson, Ruhif? Because I'm trying to find it and I am having a hard time finding it.
Jackson, if you'd like, I'm fairly certain I know which one. I can do that while you guys converse in the meantime, and I can put it in the live chat.
Send it to me. I don't want to keep delaying things. Just send it to me after we're done, because I really would want to see that.
Oh yeah,
because I've never seen that before.
Oh my goodness. Yeah, I'm terribly sorry. What is this?
Yeah, you can't find stuff when you're under pressure. You know, that's how it works on the computers. So.
Isn't that the truth? Whenever you're actually actively searching for something, it seems to actively hide from you when the moment you need it.
Been there, done that. Yes.
As have I.
Well, at any rate, as soon as I can find this, of course, it won't be, or maybe I'll find it later. But good lord. Anyways, the blog, the name of the blog, the blog is Ruhif. It's the blog Ruhif, and I can't seem to find it. Blog. It's by Glenn Williamson. Can I find it? Finally, jeez Louise. Okay, found his WordPress.
Okay. Well, anyway, I said I don't want to just hold up.
Well, here's his blog. Okay, I'm gonna put it here on the side. Here's his blog. So that's where he has all of his papers. Sequence of. But. Yeah. Okay. Well. At any rate, so yeah, it's. Let's see if I can find it. 100.
I think that maybe if
just send it to me afterward. That's fine.
Yeah,
that's fine.
It's here in the side chat. It's right here in the side chat with his blog, and so he has a whole bunch of papers on it, where you can go through. I do apologize for not being able to find it. Just offhand.
Just to avoid dead air, so we can get as many topics.
Okay.
Anyways, what other questions?
Okay, so ask your question.
Oh, now I gotta
go ahead.
remember it. I'm sorry.
Oh, that's fine.
So sorry.
I'm still looking for this thing, so no, it's fine.
It maybe wasn't that. I had a question, but it's gone now. It's maybe not important. Let's get to the audience questions.
Okay, that's fine.
Oh, very briefly for the two of you, CRISPR-Cas in the chat has suggested Idjo et al. Being the paper with the original sequence. I don't know if that's a result.
That paper sounds familiar. Idjo 2012. That sounds familiar. That's one of the papers I remember.
Anyways, yeah, we can absolutely sort it out. So,
Okay.
Anderson, if you'd like, by all means, get away. And
Are we still chatting, or are we moving on to the questions? Because
Actually, if you want, we're still gathering a few more questions. You guys can.
Okay.
We've got about
I would
10
like
more
to
minutes on the clock. If you want to keep the chat going for about 10 more minutes,
Sure.
let us finish gathering.
Oh, jeez.
So,
All
what
right.
are your thoughts then? What are your thoughts then on? Or I mean, I heard your thoughts earlier, but or sort of. When you're talking about transitional fossils, why do you not consider these as evidence for common ancestry between two groups, especially when they display characters that are intermediate between two groups?
Well, again, it depends on what you're identifying as transitional and where you're putting it at. You know, you're talking about transitional from fish to amphibian or fish to reptile? Are you talking about transition from invertebrate to vertebrate? Talking about transitional from, you know, where are we talking about transitional?
Like if we're looking, sure, we can talk about mammals. If we're looking at like the fossil record of horses and rhinos, for instance, the difference between the earliest horses and rhinos are their teeth. There's almost no difference between them skeletally. It's really just in their teeth, and so the difference is it's like rhinos are browsers, whereas horses are grazers. So why would you not consider that, for instance, as evidence that horses and rhinos are related to each other, which are common ancestors?
Yeah, but why would I have to take that as a transitional organism as opposed to just another organism that was alive at one time that's now gone? Why do I have to assume that it had some evolutionary lineage to horses and rhinos?
So is it just a separate creation? So would you say then that God made like progressively more rhino-ish animals in the fossil record?
I don't. It sounds like to me that you're imposing in. You're saying rhino-like. how much variation is there between this rhino-like and contemporary rhinos?
Well, the earliest rhinos, like Hyraceus, are these little, these small hornless mammals. So that seems to indicate a good deal of variation. And I mean, other rhinos like Paraceratherium are huge. You know,
But of course, there's
giraffes.
real small dogs and real big dogs. There's real small horses and real big horses.
So would you?
So
Okay,
size
so
is not necessarily going to be your determining factor.
but it's not just size; it's also their morphology. Their teeth are different. Whether or not they have horns is different. There are a lot of morphological
But you
characters.
just said skeletally they're virtually the same. Isn't that what you said?
There are morphological characters. There are morphological characters like having a horn versus not having a horn. Paraceratherium has this long neck. They're gigantic. They have their, I mean, yeah, and overall, they're rhinos. Certainly, they're identifiable by most of their characters, by their teeth, and those sorts of things. This is clearly a rhino. But some rhinos had, you know, a gigantic horn. Some rhinos have small horns. Some don't have horns at all. Some have two horns. So,
but aren't you describing contemporary rhinos?
There are no contemporary rhinos like Paraceratherium.
No, I mean as far as horn, no horn, large horn, small horn.
So, okay, so are all the modern rhinos descended from a common ancestor? I guess I can ask that.
Of course, I'm not the one that's going to be the most qualified to answer that, but my guess would be that would certainly be a possibility, yes.
Are there any organisms that you would say certainly are related to each other, beyond all the members of a single species?
Well, we pretty much have the wolf dog broken down genetically. We can pretty much trace all the wolf dog lineage now, starting with some ancestral wolf. So genetically, we have that down pretty good, and we know where that came from.
Right. Okay. That's great. So we can start there instead. Okay. So are wolves, the gray wolf, Canis lupus, are they related to all other wolves on the planet, like the red wolf and the maned wolf?
That's unless I'm not remembering something correctly. That's my understanding of what the genetics says. Yes.
So all wolves are related to each other, even if they're not the same genus, or the same species.
My understanding is the genetics would identify them all as starting from the same point. Yes.
Okay, awesome. Okay, great. So we agree on that. So are all members of, so are wolves related to foxes?
See, now that I'm not sure of because I don't know the genetics of foxes. So I'm going to probably say I don't think so. I'm not familiar with the fox genetics.
But I mean, they share a lot of skeletal characteristics.
They share a lot of skeletal characteristics, yeah.
Mm-hmm. But they also share a lot of genes, which is why phylogenies routinely place Canis and Vulpes as sister to each other.
And it might be, like I say, the reason I hedge is because I'm not familiar with the fox genetics. But okay, again, for the sake of argument, let's say that they are.
Okay, so if these are all related, why aren't they? Or would you say they're also related to dogs in the fossil record, like the Borophagenes and the Amphicyonids?
Which are classified as. Okay.
Sorry.
They're classified as what?
Well, Borophagenes are the bone crushing dogs. They're like these sort of souped up wolves. Kind of. They're kind of like souped up wolves. They're very large predators. They were these large caniforms, which did exist in the past, but are all now extinct. Amphicyonids is this group of these sort of dog-like animals, but rather than being digitigrade like canids are, they were plantigrade, sort of like bears are, which is why they're placed near the base, or sort of they're placed near the split between bears and dog.
All right, so let's
time.
Just I want everybody to take like a about a minute to basically recollect and basically just give like their final thoughts. Unless you guys want to go for a little bit longer, keep in mind we have a Q&A section now.
I mean, I'm ready for the questions.
All right,
But
so
I do want to say thank you, Dr. Anderson. This was fun talking with you.
Thank you.
yeah, so if you guys want to give like a 1 minute, very brief closing statement, just really quickly, just wrapping everything up, and then we'll jump into questions.
I think that the case that universal common ancestry is not true has not been demonstrated. I'm still pretty convinced that universal common ancestry is true from this. So, anyways.
Wouldn't have expected otherwise, but
Hey, I'm open to new evidence, but
I understand. We all should be, yes. So is it my turn?
Yes, it's your turn, Dr. Anderson.
Okay, all right. Well, first I want to thank Mr. Jackson for being informed. It's not much fun to debate people that aren't informed, and believe me, I've had enough of them. Boy, he gave a good explanation to differentiate what universal common descent is, and I appreciated that because I've spent debates where I wound up having to explain the evolution to the evolutionist, and so that was nice. Again, I come back to the genetics, and I understand part of what you're trying to go off of with a lot of the anatomy, but you have to recognize that, again, those are interpretations imposed on the anatomy. And one thing we find with genetics is that the assumption used to be that obviously a similar feature in two different organisms had the same genetics, and now we know they don't necessarily. The genetics has not necessarily, it's incongruities that are occurring. So it comes back again, mutation does not have the power to get you where you need to be for universal common descent.
All right, great. So it's time to jump right into the questions. First, I'd like to say thanks so much to both debaters for coming on. It's been a really fun dialogue. We'll pass it. We'll give it out to Erica to ask questions.
Actually, real quick, I'll jump into. Yeah, I want to thank both Jackson and Dr. Anderson. Really, really good dialogue. We had a really big chat today. Here's the thing: we've got so many questions. We could probably do like a 5-hour Q&A. I don't want Jackson and Dr. Anderson to feel like they need to be here all night. Did you guys want me to put a timer on how long we're doing questions for? 20 minutes, maybe.
Yes, please.
Yeah, 20 minutes is fine. That sounds fine.
Yeah, exactly. So we'll do 20 minutes, which means we'll just go through as many as possible. If, for example, let's say Jackson has a question that Dr. Anderson wanted to respond to, that's fine, and vice versa. What we like to do here is always give the last word, though, to the person who got the question in the first place. And that being said, yeah, we'll pass it over to Erica, who has gathered the questions. Thank you, Erica.
Okay, yeah, I really enjoyed that conversation, and we did have a lot of excellent questions, and everyone in the side chat and the live chat, I guess, was super involved, which is just awesome. That's genuinely what we love to see. It's what I love to see, anyways. I guess I can't speak for everyone, but yeah, it's it shows that everyone's super interested. Okay, so we're just gonna jump right into these. Stanley, would you do me a favor, and if there are any other questions while I'm reading these off, would you collect them and maybe you can read them at the end if we manage to get to the end?
Of course, yeah. And if any new super chat just came in, couple just super chats came in, just so we make sure we do get to those first, I will save those as well. So yeah, and I'll put the timer right now, 20 minutes. I'll give a 5-minute warning, and yeah, well, I'm sure this is going to fly by. So yeah, let's get it right.
So I guess I'll start with the super chat. So first from George Bond, question for Jackson Wheat. What domain of life was the last universal common ancestor if it gave rise to archaea, prokarya, or eukarya?
Well, it would have been outside of all of the domains, or as far as I understand it, all the domains that exist today are a result of it, and it would not have been one of them. It wouldn't have been bacteria, archaea, or eukarya, but it would have been a prokaryote. Very likely. It's pretty unlikely it would have been eukaryote. So there you go.
All right.
There you go. There you have it, folks.
Great. From Jeff Lynn, shalom to Gavin and all brothers and sisters in Christ. Thank you very much, Jeff, for your super chat. Those are the 2 super chats we have standing. Do you want to go and read the other super chats, and then we can just get into the regular questions?
Yes, I've got a super chat here from Logical, Plausible, Probable. Thanks so much. For JW, so that would be you, Jackson. Do you only view coding genes are evidence for evolution, or also consider non-coding regions?
Fantastic. I'm so glad you mentioned that. That's actually one of the things that I should have mentioned earlier. Gulo, the pseudogene, is actually also fantastic evidence of evolution because it's broken, but it's broken the same way in us. It has the same mutations in it that we are in us, that is also in chimps and orangutans and gorillas and the gibbons and also old world monkeys, new world monkeys, and tarsiers. It's broken in all of us the same way. We talked about it a bit in I believe it was a critique of common ancestry, a critique of intelligent design, part 1, which was about the video, a critique of common ancestry. So you can watch that, and we go into that further. So yes, I agree. It's both.
Thank you, Jackson.
Good question.
Did you want a quick response to that, Dr. Anderson, or?
Answer quick.
Yeah. Okay.
One moment.
Yeah. I'm having to refresh my memory. There's so many genes out there. Yeah. Of course, again, that involves assumptions that it's not functional, or that it's not, it's functional in some, not in others. So you're, again, making some assumptions about what the Gula gene or the family of genes actually does, that requires then that this can only come about by common ancestry, where it was, if you will, broken back in time and passed on generation to generation. So as I say, we're finding more and more function for more and more genes that were originally at one time assumed to not be functional, and even some genes that they thought they had determined were functional, and then all of a sudden, oh, I guess it's doing this after all. Because now we're finding that RNA is doing more to the messenger RNA than we ever thought, even just a few years ago. And that's coming from genes that at one time they thought they couldn't identify any function. It was just spurious RNA transcription. So I understand your argument, but you have to be careful treading into that, because you're making assumptions that involve, we simply aren't ready to say that these don't have specific functions yet.
Thank you for that response, Dr. Anderson. And Jackson, since it was your question, as we always do, you get the final word on that one.
Yeah, I hear that argument a lot, that, well, we don't know yet. And I mean, we know what it does in like all the other animals. The other organisms have, all the other animals have genes involved in the production of vitamin C. We don't. We share that trait with a bunch of other primates and our closest relatives, who do have it, or who have it, and it is functional. They also produce vitamin C. So, I mean, the evidence seems pretty clear, but I could be wrong. So there you go. I'm open to changing my mind, as always.
Okay, well, thank you, Erica. I guess I got one more super chat here, and then I'll just hand it off to you. Oh, I'm sorry. Go ahead.
No, I was just saying, yeah, no problem.
Okay, no problem. Okay, so super chat I have here that just came in from SWE. Thanks so much. Question is for you, Dr. Anderson. She asked, does Kevin understand that 2 proteins don't need the same amino acids to perform the same function? So large similarity means common descent. Go ahead.
Why does large similarity mean common descent? You just said 2 proteins don't need the same sequence to have common function. In fact, to me, that would argue against common descent, because by common descent, why would you go with 2 separate sequences if the one sequence already works? So I don't see how that is a problem for me, because that can just simply be part of what was there to begin with. It doesn't have to be based on common descent to give us 2 separate protein sequences.
Awesome. Thanks for that response. Jackson, did you have anything to say or?
Repeat the question.
Let me see real quick. Does Kevin understand that two proteins don't need the same amino acids to perform the same function?
Probably.
Okay.
I believe he does. Probably. So.
Okay. So thanks for that. Yeah, that's it for the super chats.
I don't know. I'm not going to speak for him.
I'm right at the 14-minute mark. We got 14 left for questions. Erica, it's all yours.
We have 14 left or 4 left?
Oh, 14 minutes left on the Q&A.
Okay, awesome.
You just get through as many questions as you can.
Yeah, we're going to jet through these if the interlocutors are amenable to that. So from Just a Walking Fish, can you ask Jackson why he's getting so many degrees? Is he getting the infinity gauntlet of bio to snap away creationists?
I'm getting like the normal number of degrees. I think it's bachelor's. That's what I'm about to get this fall. I hope. Fingers crossed. Then master's and then doctorate. That's pretty normal, pretty standard stuff. Yeah, right. Some researchers usually go on and get like degrees in teaching also. So
no,
some researchers do.
All right, from RJ, for both, what do you make rather of the Answers in Genesis's Ark encounter, putting transitional short-necked okapi-like giraffes and Dinohippus as the Equidbaraman ancestors?
I'll let Dr. Anderson do it. I've been answering most of these.
I will not speak for them. I mean, I'm sure they have their reasoning. You have to ask them.
All righty. Short and pithy.
I think it's interesting because was it Kurt Wise, David Kavanaugh, and Todd Wood wrote an article on the horse monobaraman where they say everything from Hyracotherium on up to the modern Equus is the same baraman. So, but then if it's all the same, or if that's all one baraman, and the difference, and as I said earlier, the difference between the earliest horses and rhinos is just their teeth. Where do we go from there? So, anyway.
Right. Cool. From David Neff, question for both. What, if anything, would convince you of evolution or creation?
You want to answer first, Dr. Anderson? Or you want me to?
Well, I come back to the genetics. That's my wheelhouse. So that's kind of my home base. And right now, evolution does not have the genetics. They don't have a mechanism. They can't explain the diversity of genes, why there are some genes that humans have that were more closely related to cows than we are to primates. There's some genes where we're more closely related to mice than we are to primates. There's some genes where elephants are more closely related to chickens than they are to other mammals. See, so you have that kind of diversity occurring, which is why Doolittle wanted all the mishmash of the tree to claim all the horizontal gene transfer occurring. So in the absence of what I consider reasonable genetic explanations for how universal common descent occurred, then I am going to maintain the position that I have.
Okay, so before I start my answer, I want to point out that Doolittle is only referring to prokaryotes and eukaryotes at the base of the tree undergoing horizontal gene transfer. Not at all about higher eukaryotes, but my answer would be, again, yeah, I'm not sure what you're referring to with elephants having genes making them more closely related to chickens. I would like to see that.
The HAR genes, the HAR genes.
HAR
There's
genes.
several HAR genes, and they vary.
You said HAR?
Around. Yeah, HAR.
HAR gene. Human is the human accelerated region. Is that what it? Human accelerated region. Is that what this is?
Hold on.
HAR gene.
I think that's what it's. I don't think so.
Because I just typed that into. Let me try typing that into Scholar. See if I come up with HAR gene.
Yeah, you're right. Human accelerated regions. Yes, that's correct.
Human accelerated regions. 49 segments of the human genome thought to be conserved throughout vertebrate evolution, but are strikingly different in humans. Oh, they are named according to their degree of difference between humans and chimpanzees. found by scanning through genomic databases, multiple species that represent loss of functions, change during evolution. Okay, well, I'll have to look these up later. Okay, I'll bookmark that one also. Okay, my answer for my answer is since it very much does look like there are transitional fossils linking groups and that genetics shows nested hierarchies. Well, at least among eukaryotes, largely horizontal gene transfer kind of screws everything up. But if there were any examples of irreducible complexity, like Darwin argued, if there were like Pegasi, whatever, I mentioned in my intro, if the genetic system were different for every Barrowman, but of course it isn't, and they fall within nested hierarchies, and that doesn't make really any sense under design. So if any of those were true, that would convince me. So
all right. Okay, from Dino Dad Reviews. Dino Dad Reviews asks, Dr. Anderson, in your presentation, did you claim that Darwin invoked magic to explain variation?
I claimed that he gave natural selection magical powers.
Okay, gotcha.
Figuratively speaking.
Figuratively speaking. I mean, he didn't actually, but because he didn't know what else caused variation, so he just assumed that natural selection does something to cause variation. And so natural selection made this happen.
Okay, gotcha. So from, oh, sorry, I don't want to cut anyone off. Jackson or Dr. Anderson, did you guys have anything else to say on that, or are we good to move on? All right.
No,
cool.
yeah.
Okay, from RJ, as a question to both, what criticism of Basner and Sanford's 2018 paper have you reviewed, and what implications might be drawn from that?
From which paper?
Basner and Sanford's.
That would be the paper where they refute Fisher's theorem.
Basner and Sanford.
It's in Mathematical Biology, 2018, volume 76, 15.
This is the waiting.
89.
This is the waiting time one.
No, this is the refuting of Fisher's fundamental theorem of natural selection.
Okay. What do I think about it? I don't think I've read this one. I don't think I have.
It simply comes down to the Fisher made wrong assumptions. Now, if his assumptions were correct, then his calculations would have been incorrect, but he made wrong assumptions, and therefore his calculations are wrong. It just comes down to that.
What does we typically see them for deleterious mutations in large populations? We typically see them come to a low equilibrium frequency, reflecting a balance between mutation and selection, but they're not doing that at high mutation rates. The essence of recombination, the key. Best on for copies of the frequency. Huh. I'm gonna have to take a look at it and look at this later. If so are they not allowing for recombination to occur in their paper?
No, I don't know why I wouldn't say that. I think that it just comes down to that they're recalculating what Fisher did based upon what we now know is population genetics versus the assumptions that he made back in the 1930s.
Well, I mean, yeah, we've certainly moved quite a ways since the 1930s. Yes, exactly. So, yeah, I mean, if he's wrong about his calculations in the 1930s, does that mean all of evolution is flawed?
No, never said that. It just simply is that Fisher has been for years offered as a mathematical proof of how natural selection drives populations to greater fitness. Well, if you remove his calculations, then you don't have that mathematical proof anymore. Doesn't mean it doesn't happen. It's just you can't refer back to Fisher.
So is there no one else who's done the mathematical calculations of it in the years hence?
You tell me.
I don't know. I'm not a big mathematics person.
Yeah, I'm not either.
I don't know. Interesting. So
we're biologists. We don't like math.
Yeah, it's okay.
All right, so moving right along for both, can you ask what the specific mechanism of lactose persistence is? And this is from Creation Myths.
What the specific mechanism is?
Yes.
Lactose persistence? I have no idea. Sure, we can look it up though. It's
you challenged that you said that this MCM6 doesn't do what I was saying it does, but of course I didn't give any explanation of what it does. The papers, the literature that has done experimentation on trying to determine lactose persistence finds that MCM6 acts in a regulatory way to the extent that if you have mutations somewhere in MCM6, you're more likely to have lactose persistence. What the exact relationship is of MCM6 to lactose is still not fully understood, but it is understood that you're knocking out some kind of regulation that typically occurs in the post-puberty adult that would, by wild type, cause them to no longer be able to produce the enzyme lactase. So something happens in the MCM6 gene that now allows the lactase gene to be continued to be expressed. So obviously you're interfering with some regulation that was there, but now a mutation has been introduced that stops that regulation from occurring.
Let's see, the enzyme lactase that is located in the villus and enterocytes of the small intestine is responsible for digestion of lactose in milk. Lactase activity is high and vital during infancy, but in most mammals, including most humans, lactase activity declines after the weaning phase. Okay. The distribution of these different lactase phenotypes in human populations is highly variable and is controlled by a polymorphic element cis acting to the lactase gene. A putative causal nucleotide change has been identified and occurs on the background of a very extended haplotype that is frequent in Northern Europeans, where lactase persistence is frequent. This single nucleotide polymorphism is located 14 kb upstream from the start of transcription of lactase in an intron of the adjacent gene MCM6. This change does not, however, explain all the variation in lactase expression. So, yeah, single nucleotide polymorphism is located 14 kb upstream from the start of the transcription of lactase in an intron of the adjacent gene MCM6. But it's not. Your argument, as I understood in the past, has been that it was a repressor. Is that incorrect? That you don't think it was a repressor?
Clearly, there is a repression going on which stops the gene from expressing the lactase, which it had been doing throughout the childhood and into puberty. So there's some kind of repression action going on. You mentioned it was cis-acting, so it's not trans-acting. So that would mean that it seems to be some kind of repressor protein that's involved. It's not just a twist of the DNA.
Yeah, but this is saying the MCM6 gene provides instructions for making part of the MCM complex, a group of proteins that function as a helicase. But so that's not a repressor, though. So whatever it's doing, I don't know, that would be interesting to find out what it is actually.
That's the thing. It may not be the MCM6. It's just the MCM6 genotype has been found consistently, as you read. Not everybody has the same genotype. But it's been found consistently to be associated with lactose persistence.
Right. Yeah. So what the argument exactly is not fully known, but it's
correct.
But it's not that this in particular was repressing per se.
Yeah. The argument is that you're taking a system that would normally be shutting down
at
the time of post-puberty that is no longer shutting down. So something that was functioning is no longer functioning. So you have lost a function, lost some level of regulation.
Yes, but does that make it a loss per se? Does that make it not necessarily a total genetic loss? Something changed in it, and you're no longer losing this ability.
Well,
wouldn't you say it was a gain because you gain this in a longer part of your life? So couldn't you flip that argument?
It would fit under what Lindsay categorizes as phenotypic pleiotropy. In other words, you lose the gain.
Okay.
So you have, you have this mutation that occurred that allows you to continue to express the lactase gene, but it was at the expense of some regulatory event that was originally occurred.
So this is saying that it contains an enhancer and an intron that increases the enhancer activity. It does not knock out a repressor. It increases its ability to compete for transcription factors. That's the mechanism of the two most common lactase persistence phenotypes. That's what I'm saying. So anyway, we got a buzzer.
Time's up. We said 20-minute Q&A. I'm not sure if Channing wants to jump in or what he wants to do.
Yeah, no, that like I said, that Q&A would fly by. Lots of good questions. Thanks so much from the audience. Lots of good dialogue in the live chat as well tonight. So I want to thank both Jackson and Dr. Anderson for giving us their time. The 2 hours has flown by. Thanks to the moderators, Erica or Guzman, if you guys had any last words.
Oh man, I just had a great time. I mean, I am going to send the remaining. I'm not sure who is going to attend which live show, but I have access to all of the, or after show rather, but I have access to all of the questions, so I'm happy to send those to John at Logical Plausible Probable if they would like to continue to go over the questions over there, because I am thinking about maybe suggesting over at my live show going over with Jackson some of the questions we didn't get to. So if Dr. Kevin Anderson would like to come to one of the after shows, that he's certainly invited to ours, and I'm sure LPPs as well. But other than that, I had a great time, and I'm really glad everybody kept things civil, and I just enjoyed listening.
Yes, me too. Thanks so much. You're absolutely right. Very civil, respectful, engaging dialogue. So we all had a lot of fun. Any last words from the debaters before we call it a night?
Thank you.
Yes, also thank you. It was nice to meet you, Dr. Anderson.
Nice to meet you.
Awesome. Well, thanks so much, guys in the chat. Shoot over to Erica's channel and John's channel for some good after shows. So I'll hand it over to the producer to